Hi guys, I am Biji and this is part 2 of NEET questions on the chapter Biomolecules. The next question is from NEET 2014. The question is, which one of the following is a non-reducing carbohydrate? And the options are maltose, sucrose, lactose, ribose 5-phosphate. Now to answer this question, first of all, you need to know what a non-reducing carbohydrate is, or for that matter, what a reducing carbohydrate is. A reducing sugar can act as a good reducing agent. But what does that mean? What are reducing agents? Reducing agents give away electrons and in the process they get oxidized. Now I shall not go into the details of oxidation and reduction. That's a whole video in itself, and probably I shall make a video on it later on. But for now, you need to know that aldehydes are good reducing agents. The CHO group can give away electrons and form the acid COOH group. And most of our sugars, they have an aldehyde group in the C1 carbon. Therefore, all these sugars are good reducing agents. Even the fructose, which has a keto group and not an aldehyde group, is also a reducing sugar. And this is because it is capable of tautomerizing such that it can create an aldehyde group in the first carbon. I shall not go into the details of it. For now, remember that fructose is also a reducing sugar. Now let's go back to the question. So is maltose a reducing or a non-reducing sugar? Let's look at its structure. So first of all, maltose is made up of glucose. Here is the ring structure of the glucose molecule. And as you can see, this is the aldehyde group, the CHO group. Now this molecule binds with another glucose molecule, forming a 1,4 glycosidic bond, which means the C1 carbon is now bonded with the C4 carbon of the other glucose molecule. The CHO group was here in the C1. Now that is a part of the glycosidic bond, and therefore this CHO group is not free anymore. However, the CHO group in this glucose molecule is still free and can act as a reducing agent. Therefore, maltose is a reducing sugar. So let's go back and look at the second option, sucrose. Sucrose is actually a 1,2 glycosidic linkage between glucose and fructose. So this is its structure. So the aldehyde group here is now involved in the glycosidic linkage. And the keto group of the fructose sugar is also involved in the glycosidic linkage, which means there are no free CHO groups here. And therefore, this is a non-reducing sugar, and that is your answer. Let's just look at lactose and ribose 5-phosphate and see how they are reducing sugars. Lactose again has a 1,4 glycosidic linkage, which means one of the sugar units will have the first carbon free, which means it is a reducing sugar. Ribose 5-phosphate, here the phosphate group is attached to the 5th carbon, which means the C1 carbon is still free, and therefore the aldehyde group there can act as a reducing agent, and therefore ribose 5-phosphate is also a reducing sugar. Let's move on to the question from NEET 2013. A phosphoglyceride is always made up of a saturated or unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is attached. A saturated or unsaturated fatty acid esterified to a phosphate group which is also attached to a glycerol. Only a saturated fatty acid esterified to a glycerol molecule to which a phosphate group is attached. Only an unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is attached. So to answer this, let's first of all take a look at a phosphoglyceride molecule. So this is your molecule with glycerol here and you have two fatty acids that are esterified to the glycerol molecule, and as you can see, they need not be saturated or unsaturated. You can have a saturated fatty acid, as you can see in fatty acid 1, and an unsaturated fatty acid. And the third molecule here is a phosphate group. That is why it is a phosphoglyceride, and it is bonded to the glycerol molecule through an ester bond. So are the fatty acids. Now coming back to the question, so your answer option will be a saturated or unsaturated fatty acid esterified to a glycerol to which a phosphate group is attached. Let's move on to the next question. Also from NEET 2013. Macromolecule chitin is sulfur containing polysaccharide, simple polysaccharide, nitrogen containing polysaccharide, phosphorus containing polysaccharide. Chitin is basically a polymer of N-acetyl glucosamine, and as you can see, it is a glucose amine. Amine meaning it has a NH2 group. So from this you should know that the answer will be option 3, nitrogen containing polysaccharide. The next question, also from NEET 2013. Transition state structure of the substrate formed during an enzymatic reaction is transient and unstable, permanent and stable, transient but stable, permanent but unstable. So you should basically just know the answer to this. It is transient and unstable. How this is so, I have discussed this in quite a detail in my video Enzymes. Do check that out. Again, a question from NEET 2013. The essential chemical components of many coenzymes are carbohydrates, vitamins, proteins, nucleic acids. The answer to this is option 2, vitamins. I have discussed this further in my video More Enzymes. Now we go on to a question from AIPMT 2012. Which of the following represents the structural formula of the basic amino acid? And the options are shown here. So what they want is a basic amino acid, which is an option for an alkaline amino acid. One of the common alkyl groups found in amino acids is the NH2 group. And if you look at these options, there are quite a few amine groups here. I shall just round that off for you. There you go. Now let's look at option 1. Option 1 has one amine group, and it is an amino acid because it has the amine group and an acid group attached to the C1 carbon. Same is with option 2. It has one amine group attached to the C1 carbon. So amino acids basically have the amine group and the acid group. And when they have a basic group and an acidic group that come together, basically the molecule on a whole is neutral because the acidity is cancelled off by the alkalinity of the amine group. So 1 and 2 are basically neutral molecules. They are not bases. Now let's go to option 3. Option 3, you can see, is not an amino acid. It does not have an acid group. So option 3 is also ruled out. Now let's go to option 4, which has to be your answer. But let's see how. As you can see, there are two amine groups. The amine group that is attached to the alpha carbon and an amine group in the R group of the amino acid. So this is a base, and this is your basic amino acid. It is basically lysine, and option 4 is your answer. The next question is from AIPMT 2012 itself, and the question is, which one is the most abundant protein in the animal world? And the options are trypsin, hemoglobin, collagen, insulin. And your answer is collagen. Collagen is a structural protein. It's a fibrous protein that is found in skin and tissues. So this is the most abundant protein in the animal world. Another question from AIPMT 2012. The organic compound shown below is a small molecular weight compound in living tissues. Identify the category shown and the component X in it. So there, that is the molecule that we have to identify, and we have to identify what X is. And the options are here. So the category and component under these, you have option number 1, which is cholesterol and guanine. And option 2 says the category is amino acid and the component is the amine group. And option 3 says it's a nucleotide with adenine as X. And option 4 says it's a nucleoside with uracil as X. If you look at the compound again, you should be able to identify it. It is a nucleoside, and therefore X will have to be uracil. It is not a nucleotide. Nucleotides have phosphate groups attached to the C5 group of the ribose sugar. So what you're seeing here is the ribose sugar with an X attached. So it is basically a nucleoside. Next question. AIPMT 2012. Which one of the following biomolecules is correctly characterized? Lecithin is a phosphorylated glyceride found in cell membrane. Palmitic acid, an unsaturated fatty acid with 18 carbon atoms. Adenylate acid is adenosine with a glucose phosphate molecule. Alanine contains amino group and acidic group anywhere in the molecule. And your answer is option 1, lecithin. It is a phosphorylated glyceride found in cell membrane. So let's go through the other options and see why they are wrong. Palmitic acid is a saturated fatty acid and it has only 16 carbon atoms. Adenylate acid is adenosine with a phosphate group, and it is not with a glucose phosphate. There is no glucose in adenylate acid. So that is also not right. And option 4 says alanine contains amino group and acidic group anywhere in the molecule. This is also not right. It has the amino group and the acidic group attached to the C1 carbon, the alpha carbon. Therefore, that is also not a right option. And the only right option here is option 1. The next question is from AIPMT 2011, and the question is, what do the X and Y axis here represent? And this is basically the graph, and they want to know what the X axis and the Y axis are. And the options given are: X axis is enzymatic activity and Y axis is pH, temperature and enzyme activity, substrate concentration and enzymatic activity and temperature. Now, as you can see, the graph is a bell curve, and you only get a bell curve when you plot enzymatic activity against temperature or pH, which means we can rule out option 3. So this cannot be a graph about substrate concentration and enzymatic activity. Now we are left with 3 other options, and as you can see, this is the optimum. As you can see, the highest activity at this point, which means the activity has to be shown in y-axis, and whatever either temperature or pH has to be shown in x-axis. The only option here in such a manner is option 2. So what you have here is a temperature optimum, and x-axis shows temperature and y-axis shows enzyme activity. Again, to remind you, I have discussed this in quite detail in factors affecting enzymes. Take a look at that video if you need to know more about this question. The next question is from AIPMT 2011, and the question is, which of the following is correctly identified along with its related function? So these are the 2 molecules that you need to identify, and the options are B is adenine, and it's a nucleotide that makes up nucleic acids. Second option is A is a triglyceride, which is a major source of energy. Third option is B is uracil, a component of DNA. And the fourth option is A is lecithin, which is a component of cell membrane. So now let's look at each of these options and see which is the right answer. B is indeed adenine. If you know the structure of adenine, you can recognize this. It is adenine. However, it goes on to say that it's a nucleotide that makes up nucleic acids, which is not true. Adenine is not a nucleotide. Adenine is a nitrogenous base, purine to be exact. Therefore, first option is not right. The second option says A is a triglyceride. Well, it is not. Let's look at the triglyceride molecule and see its structure. Yes, it has a glycerol molecule and it has 3 fatty acids attached to it. However, A also has a glycerol molecule, but it has a phosphate group that is attached to or esterified with the glycerol molecule. And further on, it has a choline that is attached to the phosphate group. So this is actually phosphatidyl choline, which is a lecithin. Therefore, your answer is 4. Let's look at option 3 and see how that's not right. B is uracil. No, it is not. We already agreed that it is adenine. But even if you do not know that, you can still rule this out because it says it is a component of DNA. Uracil is not a component of DNA. Uracil is a component of RNA. So knowing that aspect of it, you can rule out option 3. Now this question is from AIPMT 2010. The question is, which of the following statements is wrong? And the options are: enzymes require optimum pH for maximal activity. Enzymes are denatured at high temperature, but in certain exceptional organisms, they are effective even at temperature 80 to 90 degrees. Enzymes are highly specific. Most enzymes are proteins, but some are lipids. Let's look at each of these statements one by one. Enzymes require optimum pH for maximal activity. This is true. You have the highest activity for an enzyme at optimum pH. And as you move away from the optimum pH on either side, the activity declines. Enzymes are denatured at high temperature, but in certain exceptional organisms, they are effective even at temperatures 80 to 90 degrees. This is also true. Most enzymes are denatured at high temperature, but there are certain organisms, like the bacteria that grow in hot springs. For example, Thermophilus aquaticus, from which you get the Taq polymerase that is used in PCR. So this enzyme is not denatured at 80 or 90 degrees. And most of the enzymes that is present in these kind of organisms are very resistant to high temperature. The third option is enzymes are highly specific. This is also true. And the final option has to be our answer, but let's look at it. Most enzymes are proteins. This is true. But some are lipids. Now this part of the statement is wrong. An enzyme is not a protein. The only other thing it can be is an RNA enzyme. There are no lipid enzymes. So your answer is option 4. Moving on. The final question that we'll discuss today is also from AIPMT 2010. The question is, identify the components A to D in the given figure. And the figure is, yes, progress of a reaction. So you need to know what A to D is. And the options given are, option 1, A is potential energy, B is transition state, C is activation energy with enzyme, D is activation energy without enzyme. And option 2 is, A is transition state, B is potential energy, C is activation energy without enzyme, D is activation energy with enzyme. And option 3 is, A is potential energy, B is transition state, C is activation energy without enzyme, D is activation energy with enzyme. And the final option, A is activation energy with enzyme, B is transition state, C is activation energy without enzyme, and D is potential energy. This is something, as I already mentioned, that I've discussed in great detail in the video Enzymes. You should probably check that out so that you know this answer. Here I shall just give you the answer. It is option 2. A is transition state, B is potential energy, C is activation energy without enzyme, and D is activation energy with enzyme.